The next session we have is from Ines Weber of ETH Zürich. Ines graduated from ETH Zürich with a bachelor's and master's in material science and is currently a PhD student in the Department of Mechanical and Process Engineering. She has published widely in the field of sensors and catalysis and have won several awards, such as the Best Poster Award at the European Aerosol Conference. Today, we're very excited to have her share with us her work on novel gas sensors for emerging applications.
Hello, my name is Ines Weber. I'm from ETH Zürich, and today I would like to talk about novel gas sensors for emerging applications. In our lives, we rely heavily on our senses. In fact, we have put tremendous effort into translating those into electronic devices. Our smartphones, for example, can hear through microphones or voice recognition. They can see, identify faces, adjust brightness, or take images both in the visible or IR regime. And they can feel touch or identify our fingerprints. However, currently, they are not able to taste or smell. And this despite the fact that there is a huge potential in such chemical recognition. For example, in breath analysis, we could do disease diagnosis, or such detection could assist lifestyle monitoring. To put this into context, nowadays, the expenses for asthma treatment are $56 billion each year, and this could greatly be improved if we could monitor these parameters routinely in breath. Another example is air quality control. Urban population is exposed to air pollution, in fact, 96%. Routine pollutant monitoring is thus urgently needed to identify emission hotspots. A possibility to do chemical recognition or chemoresistive gas sensors. The advantage of those is that they are highly sensitive, they are compact and inexpensive. Unfortunately, however, they lack selectivity, and this is crucial as they are typically used in environments that are highly complex, such as breath or indoor air that contains more than 500 interferents. To illustrate this, I'm showing you here a sensor and a complex matrix with many molecules. Now, this sensor is not capable of detecting only one of the molecules, such as the green one. A simple approach that we can use is to implement a filter upstream of our sensor, in this case a catalytic filter, that lets only the green target analytes pass. And today I would like to show to you with two examples how we can use this approach. First, to do breath analysis and detect acetone, and second, to do air pollution monitoring to detect benzene. Let's start with the first example. Obesity is a global health challenge. As you can see here, the average body mass index is above 25 in many countries, which is considered to be unhealthy. In fact, obesity leads to more than 4 million deaths each year. It would therefore be highly beneficial if we could monitor fat burn rates. And we can do this through the biomarker acetone that forms while we are burning fat, and because of its volatility, leaves our body through the lungs and can be detected in our breath. The advantage of this is that we can do non-invasive analysis, routine measurements, and get highly personalized feedback. How do typical chemoresistive sensors respond to acetone? Here you can see one example, which is a silica tungsten sensor, and you can see that it nicely responds to acetone, but unfortunately also gives response to another analyte in breath, such as isoprene or ethanol. What happens now if we put a catalytic filter in front of this sensor? By using a platinum alumina catalytic filter, all the interferents are effectively removed and only the acetone remains, resulting in very high selectivities greater than 250. In other words, the catalytic filter here enables superior acetone selectivity. At this point, we were curious to see how this detector would perform in real breath. And for this, we conducted a pilot study with 9 volunteers that would first do an exercise protocol on a bike, and then rest for another 3 hours. You can see here exemplarily the acetone concentrations for 2 volunteers. You can see that both volunteers gave breath pulses during the first 60 minutes where they were on a bike, and then within the next 3 hours when they were resting. Most notably, we can see that our volunteer here on the left hardly changed in acetone concentrations, while the one on the right shows a steep increase, meaning that this person here burned a lot of fat and this exercise protocol was quite effective. Note that this data was measured with a high-resolution mass spectrometer that costs roughly $250,000. How does our detector perform in comparison to this? Now, luckily, our detector shows quite good agreement with a mass spectrometer, and this despite the fact that it is low cost and quite compact, as you can see here, the filter and the sensor in comparison to a €1 coin. So our low-cost detector recognizes different metabolic responses. It is also in very good agreement with a mass spectrometer, as I'm showing you here for all 9 volunteers, where you can see the sensor performance on the y-axis in comparison to the x-axis with a mass spectrometer. In conclusion, this detector could be highly promising to do personalized monitoring, for example, of exercise or dieting. Let's move on to the second example. Benzene is a global concern. In fact, accidents related to benzene have cost several hundred victims throughout the last years in India, the US, and in China. In addition, we are all exposed to benzene on a daily basis, for example, from cigarette smoke, at petrol stations, or from car exhausts. The problem with benzene is that it is carcinogenic and causes leukemia. And because of this, several organizations have set regulations into place, as you can see here. For example, in the UK, the exposure is limited to 1,000 parts per billion ppb. This is lower, for example, in the United States with 100 ppb or in the EU with 50 ppb. The strictest limit is set actually by the World Health Organization that states that there is no safe level of benzene exposure at all. Because of this, we wanted to make a device that is low cost, compact, and can detect benzene in this concentration range. So we used, again, a similar approach as before, and I'm now showing you a picture of what our detector looks like. So here you can see that, for example, indoor air would enter through the inlet and then first go through the tungsten trioxide catalyst. This catalyst here removes all the interferents except for the benzene. The benzene then carries on to a palladium tin sensor that is quite sensitive and is then analyzed with a microcontroller, which in this case is a Raspberry Pi. At the end, there is a pump that provides a continuous flow from the inlet to the outlet. And I'm showing you here another picture of the pump where it is better visible against the white background. Note that this device is portable, which is highly promising if we want to use it in a widespread environment. So how does this detector perform in real indoor air? For this, we tested it in our laboratories. Now, luckily, our laboratories do not contain benzene, so we deliberately spiked the laboratory air with benzene concentrations. And you can see here again the sensor performance on the y-axis compared to the mass spectrometer. Most importantly, there is very good agreement over the entire concentration range. To challenge our detector further, we added also additional 1,000 parts per billion of xylene and toluene, which shows deliberately xylene and toluene as they are also aromatic molecules and thus similar to benzene, and typically such chemoresistive sensors fail to differentiate between molecules within the same chemical family. But here, luckily, we can see that the xylene and toluene does not seem to affect our benzene response, and we still get quite good agreement between our sensor and mass spectrometry. Finally, I'm showing you here again the guidelines starting from 50 ppb with the EU to 1,000 ppb of the UK, and you can see that our detector nicely spans the entire concentration range. To conclude, I hope to have shown you today that selective filters and highly sensitive sensors can be combined into accurate, portable, and low-cost devices. And such devices may be promising for various applications, such as breath analysis for acetone or air pollution monitoring for benzene detection. And it is my vision that in the future, such chemical sensors will be integrated into our smartphones in a similar way that today the physical sensors are already. Thank you very much for listening.
So thank you again to Ines for that very fascinating and engaging talk on gas sensors. Similarly, we would like to invite the audience to send along any questions which they might have on Ines' work, and we'll be happy to take them as they come in. So while we're waiting for the system to populate with questions, similarly, let me just start off by asking Ines a very quick question. So Ines, it's interesting that the systems that you presented appears quite selective. Can I understand the mechanism of how the catalytic filter actually work in helping to ensure this degree of selectivity?
Yes, of course. So hello first, and thank you for your interesting question. The catalytic filter works by converting the interferents to non-responsive species. So technically, this means that the volatile organic compounds that enter the filter are combusted mostly to carbon dioxide. And carbon dioxide is inert and cannot be detected by the sensor. So this is the working principle of the catalytic filters. And ideally, the target analyte is not affected by the catalyst and can then still be detected by the downstream sensor.
Got it. Thanks so much. We do have a question from the audience, so let me read it out. And Ines, this is directed at you. So one of the audience members asked, how tunable are these devices for a different molecule?
This is an excellent question, thank you. So if we want to look at other volatiles, for example, we definitely need to choose another catalyst. I could see that during my research work, when we changed the catalytic filter, also the selectivity changes drastically. And here my advice is to look closely into heterogeneous catalysis that already gives a very broad database or broad materials. And there we can see what materials may be preferentially reacting with the target analytes or with the interference.
Great. We do have a couple more questions, so very fascinating. Let me read off one other question that came in. So an audience member says that it's a really nice talk, and he's asking if you can please elaborate on what kind of new and ideal materials will be promising in acting as sensors.
Yes, thank you for the question. In terms of sensing, in my research work, I found that the problem of the sensor alone is typically the selectivity and not the sensitivity, meaning that when we take even a palladium tin or a tin oxide sensor, which is the most common one in terms of metal oxide sensors, it is usually capable of detecting low concentrations, but it cannot do this selectively. So I think that emerging materials may be used also for sensors. There's a lot of examples also, such as tungsten is used or indium is used. But if they can really be very selective in a way that they are when combined with a filter, I'm not quite sure if this is possible. So I think the material choice of the sensor is important in terms, but not necessarily in terms of selectivity.
Great, very important distinction there. Yeah, thanks, Ines. One more question that just came in. Very interesting work. Have you also tried detection of volatile chemical warfare agents with your system, like nerve agents?
I'm sorry, I'm not sure if I completely understood the detection of which one.
Sure, let me repeat it. So this question is quite interesting. It's asking if you have tried detecting warfare agents. So for example, your nerve agents.
Oh, now I understand. I have not actually. Yes, interesting point. I have not tried that, but I think that should be possible as well. There we would have to see how it responds to the sensors. In principle, I think that should be doable. Again, we just need to look at the analytes that we're interested in, how they react with the specific materials, both the sensor and possibly a filter. But I have no experience, but it would be interesting to look into that. Thank you for the idea.
Yes, we have a very we have a great audience with very creative minds here. So one more question, which again is quite relevant when you talk about sensors. And here, this question is: how long can your sensor system work before the function expires? So it's a question around the lifespan of these the platform that you have developed.
Indeed, a very important question. I have tested my filters over the time of 150 days, so several months where I did not see any degradation. And I will carry these measurements on. So far, I have not seen how they perform after a longer time, but I think that they can be quite stable, at least in a laboratory environment as we have it here. The question is then, of course, if they're extensively used and also exposed to highly concentrated or other scenarios, that may change. But the way that we operate them here in our laboratory, we have quite good stability over months.
Great to know. One final question, Ines, that just came in. What are the limitations of the catalytic filter? Are they difficult to produce?
So the limitation, I think, is not the production. Here in our lab, we use flame spray pyrolysis, but even commercial catalytic filters could work. I think one of the limitations is that it's very, very difficult to fabricate a filter that has the selectivity that we desire. So to remove all the interference, but not remove the target analyte. It's very difficult to shift this, especially if we want to distinguish between molecules within the same chemical family or with a similar chemical structure. So I think this is the most challenging task about the catalytic filters.
Thank you. So yeah, thanks again, Ines, for that very engaging presentation, and also to the audience for a very engaging discussion. So thank you,
very
Ines.
much.